Welcome to Crisis in Perception, where we examine the systems shaping our world.
And really looking at the hidden architectures behind the things we just take for granted.
Exactly. And today's deep dive is incredibly special. If you are tuning in, we know exactly who you are. You are the learner.
You're someone looking to cut through the noise.
Right. You want to grab the essential foundational insights without suffering from that constant info overload we all get. You want the deep knowledge, but you need it structured, engaging, and directly applicable to how you understand the world.
Which is exactly what we're aiming to deliver today.
Right. So today, we are exploring a topic that touches the very core of human existence, the future of healthcare, and really, the actual limits of human perception. Modern medicine makes this incredible promise to you, the promise of ultimate personalization.
The idea that your healthcare can be tailored to the exact specifications of your DNA.
Right, but that massive promise hinges entirely on a single question. Can we actually interpret biological complexity at an unprecedented massive scale?
And that framing hits the core of the issue perfectly, I think. We are looking at a fundamental shift in how science operates. For decades, the primary bottleneck in understanding life was physical data collection.
Getting the data in the first place.
Right. It was about engineering the physical machines that could simply read the letters of a genome. I mean, the Human Genome Project took over a decade and billions of dollars just to get one draft.
Which is wild to think about now.
It is, because we have largely solved that hardware problem. The bottleneck in understanding life is no longer data collection. The crisis we face now is a crisis of perception.
A crisis of perception. Huh?
Yeah, the bottleneck is pattern recognition itself. We have oceans of data, but our traditional statistical tools simply cannot perceive the biological patterns hidden within it.
So to explore this crisis of perception, we have a really fascinating stack of source material today. We are pulling from the newly published 2026 book, Applications of Deep Learning in Genomics.
Edited by Bipin Kumar Rai, Rupa Rani, and Gautam Kumar.
Right, published by CRC Press. It is a comprehensive look at how artificial intelligence is colliding with biological sequence data. And our mission for this deep dive is not just to look at AI as some magic, standalone breakthrough that cures diseases with the push of a button.
Because treating AI as magic is the fastest way to misunderstand what is actually happening in modern science.
Exactly. We are going to unpack this as a layered technical system where biological complexity, computational architecture, massive data infrastructure, and institutional incentives all sort of crash into each other.
The system design matters immensely here. Biological knowledge is increasingly being filtered through computational architectures that dictate what becomes visible, what becomes measurable, and ultimately what a doctor can act on in a medical clinic.
Okay, let's unpack this from the foundation. Let's start with the sheer scale of the biological data we are dealing with. Because when someone says the word data, I think the average person, pictures a large Excel spreadsheet.
Right, maybe a few thousand rows.
Yeah. But that doesn't even begin to cover what we are talking about with modern genomics, does it?
Not at all. A spreadsheet is entirely the wrong mental model. To grasp the scale, we have to look at the traditional methods of computational genomics and how high-throughput sequencing technologies changed the paradigm.
So we're talking about next-generation sequencing, or NGS.
Exactly. NGS and single-cell RNA sequencing. Before these technologies, sequencing was slow and highly targeted. You looked for a specific gene. But NGS allows researchers to sequence entire genomes, transcriptomes, and epigenomes simultaneously and at incredibly high resolutions.
And the speed is what's really staggering, right?
Oh, it's unbelievable. You can sequence a complete human genome in roughly 5 to 6 hours today.
5 to 6 hours.
Yeah.
For the entire code of a human being? But the result of that speed is a complete data deluge.
A flood of information.
Yeah, the sources mention that a single human genome yields somewhere between 100 to 150 gigabytes of raw data. And that is just for one individual. If a university is running a large population study trying to find genetic markers across 50,000 people to understand a complex disease, they're instantly generating petabytes of information.
Petabytes. And volume is only the most superficial layer of the problem.
Okay, so what's the deeper layer?
If we connect this to the bigger picture, the real issue, the first layer of our complex system is the biological complexity within that volume. This data is highly dimensional and deeply messy. A single biological sample contains millions of interacting variables.
So you're not just looking at a static string of DNA letters, the A, C, T, and Gs.
Exactly. You are looking at RNA expression levels, protein interactions, and epigenetic marks all at once.
Let's actually define our terms there, because It is so easy to get lost in the jargon. For someone listening whose biology knowledge maybe ended in high school, what exactly are we measuring when we talk about RNA expression and epigenetics alongside the DNA?
Sure. Think of the DNA as the master blueprint for a massive factory, locked securely in the architect's office. It contains the instructions for absolutely everything, but it doesn't actually do the building.
It's just the reference material.
Right. When a cell needs to build something, say a protein, it makes a temporary photocopy of a specific section of that blueprint. That photocopy is the RNA.
Right.
So measuring RNA expression, which we call the transcriptome, tells us which specific blueprints are actively being read and utilized by the cell at that exact moment.
So DNA is what you can do, and RNA is what you are currently doing.
That's a great way to put it.
What about the epigenetic marks then?
Epigenetics are the chemical modifications that sit on top of the DNA. They don't change the underlying ACTG sequence at all, but they control whether a gene is turned on or off.
Like a physical switch?
Kind of. For example, the DNA is wrapped around protein spools called histones. If the spool is wound very tightly, the cellular machinery simply can't read the blueprint. It's hidden. But if it's wound loosely, it can be read. The dataset captures all of this simultaneously. It captures the underlying sequence, the chemical tags telling the spool to wind or unwind, and the volume of RNA photocopies currently circulating.
So when you feed all of that into a computer, how do these variables actually interact? Are they acting in simple, predictable pathways? Like gene A causes thing B.
Not at all. That's the problem. The biological relationships are fiercely nonlinear. Genes act in combinatorial ways. The expression of a single gene might depend entirely on the simultaneous presence of 3 other genes, plus a specific epigenetic marker on a totally different chromosome.
And maybe an external environmental factor, too.
Exactly. This is where traditional statistical models completely break down. You cannot run a standard linear regression analysis on petabytes of interacting, multi-layered data and expect to find the exact mechanism for Alzheimer's disease.
It's just too complex.
Basic algorithms simply cannot perceive the patterns hidden inside this level of heterogeneous noise.
To put an analogy to this for the listener, it feels like being handed a library containing 100 million books. But all the pages have been ripped out, thrown into a massive pile on the floor, and covered in ink spills and typos.
That's a very accurate picture of raw genomic data.
Right, and to make it infinitely worse, the books are written in a strange code, where the meaning of a specific word actually changes, depending on which book was sitting next to it on the shelf before the pages were ripped out.
Yes.
You have all the information in the universe, but no human brain or simple equation can actually read it.
That analogy perfectly captures the crisis. The information is technically there, right there in the pile. But the spatial and temporal dependencies are too vast. The signal is completely buried under a mountain of noise.
So how do we actually start reading this ripped-up, noisy library? If human brains and standard statistics fail, this is where the computational architectures have to step in.
Exactly.
Which brings us to section 2 of our deep dive, the architectures of perception. How AI actually reads the code of life.
We have to understand that biological sequences, whether they are DNA, RNA, or chains of amino acids making up a protein, are conceptually just data with deep dependencies.
Okay.
Each nucleotide has a specific location, and its relationship to the other elements in the sequence contains the critical information. Deep learning provides powerful neural network architectures that can automatically extract those features from the raw data.
Automatically, so without a human stepping in.
Right, without requiring a human to manually construct the rules or hypotheses first.
That feels like a massive paradigm shift, because traditional science is human-directed hypothesis testing. The scientist says, I think gene A causes disease B. Let's run the data to check.
Yes, the human leads.
But this is machine-directed pattern extraction. The machine just looks at the raw data and says, here are the relationships, regardless of whether you suspected them or not.
It fundamentally flips the methodology. The deep learning model makes assumptions about the underlying biology based purely on the mathematical distribution of the data itself.
Okay, so how does it actually do that?
Well, the book breaks down several specific types of neural networks that perform this translation. Each architecture has a different reading style, optimized for different types of biological patterns.
Let's walk through them, because this is where the actual mechanics of the AI come into play. First up, the text focuses heavily on RNNs, recurrent neural networks.
Yes, and their more advanced iterations, LSTMs, which stands for long short-term memory networks.
What is the specific reading style of an RNN?
RNNs are specifically designed to process sequential data where the order of the information matters immensely. Think of the ATCGs of a DNA strand. The sequence order is the entire point.
Right. If you scramble the letters, it's meaningless.
Exactly. Unlike older networks that treat every input independently, RNNs process data sequentially, step by step, and crucially, they maintain an internal state, a hidden memory.
So they remember what they just read.
Yes. As they read the second letter, they retain mathematical context from the first letter. This allows them to keep track of the dependencies between elements in a sequence.
But I've read that traditional RNNs have a major flaw when it comes to really long sequences. They suffer from something called the vanishing gradient problem.
Yes, they do.
What does that actually mean in a biological context?
The vanishing gradient problem essentially means the network has a short attention span. As the RNN processes a very long sequence, the mathematical influence of the earliest inputs gradually fades away to zero. It literally forgets what happened at the beginning.
And in genomics, that's a problem.
It's fatal. You might have a regulatory element, an enhancer, that affects a target gene tens of thousands of base pairs away on the DNA strand.
Right.
That is a long-range dependency. A standard RNN would lose the connection before it finished reading the distance between them.
And that is where the LSTM comes in to fix the memory issue.
Yes. LSTMs are a sophisticated variant of RNNs. They are equipped with specialized mathematical gates, specifically an input gate, an output gate, and a forget gate.
A forget gate.
Yeah, these gates actively regulate the flow of information, deciding which data to keep in long-term memory and which irrelevant noise to discard.
So it's actively curating its own memory.
Exactly. This allows LSTMs to effectively model those crucial long-range dependencies over vast genomic distances. They are currently used for complex tasks like forecasting RNA-binding protein interactions and modeling how gene expression changes over time.
Okay, so if RNNs and LSTMs are sequential readers, moving left to right and remembering the context, what about CNNs? Convolutional neural networks.
CNNs are fascinating here.
Because outside of genomics, CNNs are famous for image processing. They are the engines behind facial recognition and self-driving car vision systems. How does an image processor help us read a string of biological text?
It requires a very clever adaptation. In computer vision, a CNN uses a two-dimensional grid to scan an image for spatial hierarchies. It looks for edges, which combine into shapes, which combine into a face.
Right, a 2D scan.
In genomics, researchers adapted them into one-dimensional convolutions. Instead of a 2D grid sliding over pixels, a 1D filter slides across the biological sequence.
Like a sliding window moving over the text.
Precisely. And as this window slides, it isn't trying to remember the whole sequence from left to right like an RNN. It is scanning for local spatial hierarchies.
What does that look like in DNA?
It is looking for conserved motifs or highly specific functional regions. For example, it might be trained to recognize the exact shape and pattern of a transcription factor binding site.
Which is what?
The specific landing pad on the DNA where a protein attaches to turn a gene on. The CNN automatically learns the local characteristics of that sequence regardless of where it appears in the entire genome.
So if we go back to our library analogy, if you are trying to understand a massive biological document, the RNN is reading the sentence left to right to understand the grammar.
Yes.
But the CNN is like a researcher with a highly trained magnifying glass, sliding quickly over the pages, ignoring the grammar entirely, and just looking for a very specific visual phrase or motif that pops out.
That is a very effective way to distinguish their utility. However, the current landscape of deep learning in genomics is being dominated by a newer, far more powerful architecture.
The heavyweights?
The heavyweights of the field are now transformers and attention mechanisms.
Transformers. This is the underlying architecture behind the massive large language models that have basically taken over the world recently.
Yes, like ChatGPT.
Right. And the text mentions adaptations of BERT specifically for biology. Why are transformers replacing RNNs?
Transformers revolutionize sequence evaluation because they completely discard the sequential left-to-right processing requirement of RNNs.
Oh, they don't read left to right.
No. An RNN has to wait to read letter 100 until it has finished reading letters 1 through 99. Transformers process the entire sequence in parallel, simultaneously.
Right.
And they achieve this through an innovation called the self-attention mechanism.
How does self-attention work when applied to biology?
Self-attention allows the model to look at a single nucleotide in a sequence and simultaneously calculate a mathematical weight and attention score representing its relationship to every other nucleotide in that entire sequence,
Regardless of physical distance.
regardless of distance. It essentially asks, how much does this specific A matter to the G, 1,000 bases down the line? It understands both the local motif and the global long-range dependency at the exact same time.
So the transformer isn't reading left to right, and it isn't just sliding a magnifying glass.
Yeah.
To push the analogy, it is looking at the entire 100-page book at once in a single glance and instantly understanding how a slight typo in the prologue fundamentally alters the climax in Chapter 50.
That parallel processing capability is exactly why they're so powerful. It makes them incredibly efficient at handling the massive sequences generated by whole genome sequencing.
Because it can see the whole board.
Yes. Because they map the entire context simultaneously, they are unparalleled at identifying new genes, classifying complex genetic variants, and predicting how a single point mutation will alter the 3D folding structure of a protein.
Now, the text also dedicates space to autoencoders. These seem to serve a slightly different function than the predictive models we just discussed. They are described as unsupervised models. What role do they play in the architecture of perception?
Autoencoders are brilliant mechanisms for dealing with the noise and dimensionality we discussed in Section 1. An autoencoder is structured structurally like an hourglass.
An hourglass, okay.
It takes the high-dimensional, messy, raw data and forces it through a mathematical bottleneck. This is the encoder phase. This compresses the data into a lower dimensional space.
Okay, so it squishes it down.
Then the decoder phase attempts to reconstruct the original data from that compressed representation.
Wait, let me pause there. Because if you compress it and then blow it back up, aren't you losing data? Why would you intentionally put your data through a bottleneck?
Because the bottleneck forces the network to learn only the most essential underlying representation of the data. It simply cannot memorize the noise. It only has enough capacity to remember the core structural patterns.
Oh, that's incredibly clever.
It is. In genomics, researchers use specific variants called denoising autoencoders. You purposefully feed the model corrupted, noisy genomic data and train it to reconstruct a clean, pristine version.
Like taking a blurry photo and making it sharp?
Exactly. The autoencoder effectively filters out the technical measurement errors and the biological static, leaving you with a highly accurate representation to pass on to your transformers or CNNs.
Which is absolutely vital, because before we can use a transformer to predict a disease or design a custom drug, we have to actually possess a clean, accurate copy of the patient's DNA.
Right. You can't predict on garbage data.
And as the book explains, getting that clean copy is a massive computational hurdle on its own. That leads us directly to Section 3 of our deep dive, reconstructing life and the mechanical pipeline of DNA fragment assembly.
This is where the theoretical architecture meets the physical reality of the laboratory. We mentioned next-generation sequencing earlier.
Right, the fast sequencing machines.
The physical reality of these high-throughput machines is that they do not and physically cannot read a human genome from start to finish in one long, continuous string.
The machine just can't do it.
No, the chemical processes break down if it gets too long. Instead, they shatter the DNA into millions and millions of tiny fragments, read those tiny fragments, and spit out millions of short reads.
So rebuilding the original continuous sequence from those millions of tiny text fragments is what the field calls fragment assembly.
Yes, and it is arguably the most computationally intense puzzle in modern science.
I want to visualize this puzzle because the sheer scale is hard to comprehend. Imagine assembling a billion-piece jigsaw puzzle.
A billion pieces?
Right. But to make it accurate to genomics, there are two massive complications. First, the human genome is highly repetitive. Almost half of our DNA consists of repeated sequences.
It's a lot of repetition.
So in our puzzle, millions of the pieces are identically blue sky. Just identical. Second, the sequencing machines aren't perfect. They glitch.
Right.
So some puzzle pieces have the wrong shape cut into them or the wrong shade of blue printed on them. How did researchers solve this puzzle before AI?
Well, they relied on traditional computational algorithms. The text highlights a few foundational methods: De Bruijn graphs, overlap layout consensus, which is known as OLC, and alignment search tools like BLAST or Smith-Waterman.
Let's define those, because this is where the jargon usually loses people. If I'm a researcher in 2010 trying to build this puzzle, what is a De Bruijn graph actually doing on my computer?
A De Bruijn graph is an elegant mathematical concept. Instead of trying to compare whole puzzle pieces, the algorithm takes every short read and shreds it further into even smaller, overlapping chunks of a fixed length.
Okay, making the pieces even smaller.
Yes, these are called k-mers. It then creates a massive network graph, drawing a path from one k-mer to the next based on their overlaps. By tracing the most logical path through this immense network of nodes, it attempts to spell out the original sequence.
Interesting. And what about OLC? Overlap, layout, consensus.
OLC works a bit differently. It directly compares every single read against every other read to find where the ends overlap.
That sounds computationally heavy.
Extremely heavy. Once it finds all the overlaps, it builds a layout graph showing the relative positions of all the fragments. Finally, it calculates a consensus.
Meaning what?
Essentially taking a vote among all the overlapping pieces to determine the final sequence of letters. In tools like BLAST's basic local alignment search tool function almost like a search engine.
Like Google for DNA?
Kind of. They use heuristic algorithms to rapidly find regions of local similarity between a read and a known reference genome.
But if these traditional algorithms have been the foundation for over a decade, why are we replacing them with deep learning now? What is the breaking point?
The breaking point is the exact complications you mentioned in your puzzle analogy. Repetitive regions and sequencing errors.
The blue sky pieces.
Yes. When a de Bruijn graph hits a massive section of identical blue sky puzzle pieces, the repetitive genomic regions the graph tangles into massive, unresolvable loops. It simply doesn't know which way to go. It gets stuck. Furthermore, these classical algorithms are highly sensitive to the sequencing errors produced by the machines. A base substitution error causes the graph to branch off incorrectly.
Which leads to what?
The result is often a misassembly. The genome is literally put together wrong.
And a wrongly assembled genome is completely useless and potentially dangerous if you were trying to use it to make precise medical decisions.
Exactly.
So how does deep learning intervene here? The text outlines 3 key AI interventions in the assembly pipeline. Let's start with error correction.
Error correction is where deep learning really shines. Machines like Illumina sequencers produce noisy reads with specific types of errors. Base substitutions, where an A is read as a C, or insertions and deletions, where a letter is added or skipped entirely.
Just machine typos.
Right. Traditional statistical tools try to fix this by looking at frequency, but they often fail. Deep learning models, specifically CNNs and RNNs, take a totally different approach.
How so?
They are trained on massive data sets of DNA sequences where the flaws are already known. They learn the contextual patterns of the sequencing machine's specific errors.
So the model looks at the fragment and says, the machine printed a C here, but based on the complex context of the 20 letters surrounding it, the machine historically glitches in this exact scenario. That C is definitely supposed to be a G.
Exactly. They fix the puzzle pieces before you even attempt to put them together.
That's incredible.
The second intervention is overlap detection. As we discussed with the OLC method, finding where fragments overlap is computationally grueling. Comparing every piece to every other piece in a data set of billions takes massive server time.
Right, because the math scales up exponentially.
Deep learning models, particularly attention-based transformers, can circumvent this entirely. They are trained to recognize the implicit mathematical pattern of overlapping sequences without having to perform the explicit, computationally heavy alignment step.
Did you see the pattern?
They look at the properties of 2 sequences and rapidly predict the probability and location of an overlap.
It literally shortcuts the math. It just recognizes the fit based on the pattern. And the final step is fragment merging and post-assembly refinement.
Yes. Once the overlaps are detected, the fragments are merged to create longer contiguous sequences called contigs. But because of the repetitive regions, misassemblies still happen.
So the AI acts as quality control.
Exactly. This is where AI performs QC. CNNs can scan the draft assembly, look at the depth of the sequence coverage and the spatial patterns, and actively flag regions that look structurally incorrect.
And transformers can help there too.
Yes, transformers can analyze the long-range dependencies across the entire draft to ensure the biological logic actually holds up. If it spots a flaw, the AI can suggest targeted fixes, like reassembling a specific loop or using predicted sequences to bridge a gap.
But hold on, let me play devil's advocate here for a second.
Sure.
If the machine's initial read is a C, and the AI model uses context to change it to a G, or if the AI is actively broadening gaps in the assembly, isn't there a massive risk that the AI is hallucinating a fix that isn't biologically real?
That's the main fear, yes.
How do we know it's not just making up data to make the puzzle fit its own mathematical assumptions?
That is a highly critical point, and it is the exact reason why researchers do not blindly trust the AI. Deep learning models provide probabilities, not absolute certainties. The model assigns a confidence score to its correction. If the confidence is low, the original data is retained. Furthermore, the AI's output is constantly cross-validated against physical, long-read sequencing technologies, like those from Oxford Nanopore.
And what do those do?
They provide lower accuracy on the individual letters, but much longer physical context. So we use the AI to refine the short reads, not to invent biology out of thin air.
Which leads perfectly into the system constraints the book discusses. The authors strongly advocate for hybrid strategies. We aren't throwing the classical algorithms in the trash.
Not at all. Standard algorithms like the Bruin graphs have been mathematically proven and refined for over 2 decades. They are incredibly robust at building the initial rough draft of the genome.
So they work together.
The hybrid approach uses those classical algorithms to do the heavy lifting of the initial graph traversal and contig generation. Then, it deploys the deep learning models to perform the targeted detail work.
Fixing the complex sequencing errors.
Right, resolving the tangled loops in the repetitive regions and polishing the final alignment.
It's a structural partnership. The old algorithm builds the raw scaffolding, and the deep learning model comes in to fix the structural flaws and finish the interior.
Exactly.
But this raises a broader question. If this hybrid AI approach is so powerful and solves all these problems, why isn't every lab in the world running it right now?
Because we are dealing with a complex physical system, and systems have material constraints. This transitions us from the theoretical code to the actual physical infrastructure of modern science.
The real-world costs.
Right. We have to look at Section 4, the incentive architecture and the structural realities of compute, data, and power.
Because as we dive into this layer, it becomes brutally clear that deep learning is not cheap, and it is certainly not democratized.
If we look at this through a systems lens, we see a very specific data model institution pipeline. The first massive material roadblock is scalability and compute power.
Right.
We just talked about processing petabytes of high-dimensional genomic data using transformers and CNNs. That is incredibly computationally demanding.
You can't just run this on a laptop.
Oh, absolutely not. It requires immense hardware resources for both training the models initially and running inference on new patient data.
The book notes that processing this scale of data requires multi-GPU training clusters. You have to use distributed computing frameworks like TensorFlow and PyTorch, and you are almost entirely reliant on massive cloud platforms like AWS or Google Cloud just to have the server space to run the math.
And renting that server space costs an exorbitant amount of money. The computing cost rises exponentially with the size of the datasets and the parameters of the models.
So smaller labs are just priced out.
Completely. But the second and arguably more critical structural roadblock is the data requirement itself. Deep learning models are structurally data hungry. To achieve high predictive accuracy, they require massive volumes of perfectly labeled, high-quality training data.
We're talking about massive repositories like GenBank or the Cancer Genome Atlas,
Right.
which take decades, international cooperation, and hundreds of millions of dollars to compile and annotate.
Exactly. And the data must be vast, because if a complex deep learning model is trained on a small or incomplete dataset, it suffers from a mathematical failure called overfitting.
Let's explain overfitting clearly for the listener. Overfitting is when the model essentially memorizes the answers to the specific test it was trained on, rather than learning the underlying biological rules.
Right? That's a great way to think of it.
It's like a student who memorizes a specific practice exam word for word. They get 100% on the practice test, but when you give them the actual final exam with different questions, they completely fail because they didn't actually learn the subject matter.
That is a perfect explanation. The model's internal parameters become too tightly tuned to the specific noise and quirks of the training data. When it encounters real-world patient data that deviates even slightly from that specific distribution, it fails to generalize.
So breaks in the real world.
It fails clinically. To avoid overfitting, you need massive, diverse datasets. And this requirement creates a profound structural reality. Predictive authority concentrates in very specific places.
It concentrates in data-rich institutions.
Yes. The major tech companies and the massively funded legacy research universities, they are the only entities that can afford the GPU clusters and hold the licensing rights to the petabytes of labeled data.
So the system is pushed towards scale and automation by the entities that have the resources to build the pipeline.
Right.
But this mathematical requirement for massive data introduces a severe clinical risk. It is the risk of out-of-distribution failure.
This is critical.
If the massive datasets used to train these models predominantly feature genetic data from specific demographic populations, and historically, genomic databases are overwhelmingly skewed toward populations of European descent, then the model learns the statistical distribution of those specific populations incredibly well.
Very well.
But if you apply that model to a patient whose biology or ancestry was underrepresented in the training data, the model mathematically struggles.
It struggles because the patient's biological data falls outside the mathematical distribution the model was optimized for. It literally hasn't seen those specific combinatorial interactions before, so its predictions become far less accurate.
This is a crucial point for you, the listener, to grasp regarding the promises of modern healthcare. The entire field is marketing precision medicine. The promise that a drug or a diagnostic tool will be tailored to your specific DNA.
Right.
But if the structural pipeline, the datasets used to build the AI, did not include your specific biological variations, then that model might overfit to someone else's biology. The resulting medicine might not be very precise for you at all.
It is a fundamental limitation of data-driven systems. And this structural push for automated predictive authority leads directly into the most intractable bottleneck when it comes to clinical application.
Which brings us to Section 5.
Right, Section 5, the interpretability crisis.
The black box.
The black box. Deep learning models, particularly deep neural networks and transformers, are inherently opaque.
Meaning we can't see inside them.
Right. A model can analyze 1 million variables across a patient's multi-omics profile and accurately predict with 99% certainty that the patient will develop a specific autoimmune disease. But the model cannot explain the underlying biological logic of how it arrived at that conclusion.
I'm struggling to see how that works in practice. If it's 99% accurate, why does it matter if we know how it did the math? If a machine tells a doctor, this patient has a 99% chance of heart failure, shouldn't the doctor just act on it?
Because in medicine, accuracy is not enough. You need mechanistic transparency for both clinical liability and biological advancement. Doctors rely on clinical decision support systems, or CDSS.
Right.
But a doctor cannot legally or ethically act on a recommendation to perform a drastic surgery or prescribe a severe chemotherapy without knowing the biological why behind the prediction.
That makes sense.
If the model says administer this toxic drug and the doctor asks why, which specific gene pathway is malfunctioning, the AI essentially outputs a shrug. Its internal logic is distributed across millions of abstract mathematical weights.
So we have built an oracle that gives perfect answers, but speaks in a language we can't decipher.
Exactly.
And that creates a massive barrier to adoption. Doctors won't use a tool they can't validate. We assume that because an AI can predict something better than a human, it must understand the biology better. But that is a total misperception.
It is the defining misperception of the current era of AI in science. We constantly conflate predictive power with mechanistic understanding.
They aren't the same thing.
No. If a model accurately diagnoses a disease, but we do not know the biological pathway it utilized, we haven't actually advanced human understanding of the disease. We have simply outsourced our medical authority to an opaque mathematical abstraction.
We know that it works, but we are blind to how it works.
Exactly.
So how is the field attempting to open the black box? The book mentions several post hoc explanation methods designed to shine a light on the model's internal logic.
The field is heavily investing in interpretability tools. The text highlights saliency maps, which generate heat maps over the input data to show which regions activated the network the most.
Visualizing the attention.
Right. It also highlights the attention mechanisms we discussed earlier. In a transformer model, the attention weights can be extracted and visualized to show exactly which segment of a genetic sequence the model was focusing on when it made its prediction.
So the model essentially takes a digital highlighter pen and says, I made this cancer diagnosis because my attention was heavily weighted on this exact string of 50 letters right here.
Yes. But the most prominent tools discussed in the text are post hoc methods like Esche and Lime.
Let's walk through the real-world mechanics of these because they are crucial to the future of the field. What is SAP actually doing?
SAP stands for Shapley Additive Explanations. It is a concept borrowed directly from cooperative game theory.
Game theory.
Yeah. Imagine a team of workers building a product. In our case, the workers are the features in the data gene A, a specific epigenetic mark, and the patient's age. The product is the AI's final prediction. SHAPE calculates the marginal contribution of each individual worker to the final product by systematically testing the model's output across every possible combination of those workers.
It removes gene A, runs the model, sees how much the prediction drops. It puts gene A back, removes the epigenetic mark, runs it again.
Exactly. Through exhaustive combinatorial math, it assigns a specific numerical value to every single feature.
So it quantifies the blame or credit.
Right. In a clinical setting, if the AI predicts a high risk of heart disease, a SHAPE tool doesn't just give the doctor the raw percentage, it outputs a visual breakdown. Gene variant X increased the risk by 40%, the patient's age increased it by 10%, but their expression of gene Y actually lowered the risk by 5%.
That is incredibly useful. And what about LIME, local interpretable model-agnostic explanations?
LIME takes a different approach. Instead of trying to explain the entire massive model, LIME focuses on local perturbation.
What does that mean?
If the AI makes a specific prediction for patient A, LIME takes patient A's data, slightly alters it, tweaks a gene expression value up or down, and feeds it back into the model to see how the prediction changes.
Just poking it to see how it reacts.
Yes. By doing this thousands of times around that single specific patient, it builds a simple, understandable linear model that approximates how the complex AI made its decision in that localized instance.
Both of these tools act as translators. They force the opaque math to break down its decision into biological threads that a human doctor can actually pull on and validate in a physical lab.
They are critical bridges between mathematical pattern recognition and biological mechanism, because until the black box is transparent, the clinical integration of these powerful systems will remain fundamentally stalled.
Okay, so we've covered a massive amount of structural ground: the data deluge, the architectures of perception, the fragment assembly puzzle, the monopolies on compute power, and the interpretability crisis.
It's a lot of constraints.
It is. But let's pivot slightly from the structural constraints to the incredible world-altering potential when these models actually are successfully applied. Let's look at Section 6, Precision Medicine and Deep Reinforcement Learning.
This section is where we see the transition from theoretical genomics to active clinical intervention.
Real-world medicine.
Right. The ultimate goal of the system is discovering novel disease biomarkers, predicting how an individual patient will respond to a specific treatment protocol, and stratifying patients to create truly customized therapeutics.
And Deep Reinforcement Learning, or DRL, is uniquely suited for this dynamic challenge. How is DRL structurally different from the neural networks we've been discussing, like CNNs or transformers?
DRL represents a completely different paradigm of machine learning. CNNs and transformers generally learn passively from static historical datasets.
They just read the books.
Right. DRL is an active learner. It learns via trial and error in dynamic environments. It operates on a framework of 3 components: an agent, an environment, and a reward system.
Okay, break that down.
The agent interacts with the environment, takes a specific action, observes the result of that action, and receives a numerical reward or penalty. Over millions of iterations, it maps the environment and figures out the optimal sequence of actions to maximize its cumulative reward.
That sounds like how you train a dog, or how an AI learns to play a video game like Mario.
Exactly the same concept.
But how do you apply a trial and error agent to a biological environment? You can't just let an AI run trial and error experiments on a live human patient.
Absolutely not. You do it entirely in simulation. And the primary application is drug discovery and virtual screening. We have to understand how agonizingly slow traditional drug discovery is.
Decades, right?
Yeah. You have a biological target, say a mutated protein causing a tumor. Traditionally, pharmaceutical researchers empirically test thousands of physical-chemical compounds in a wet lab to see if any of them bind to that target. It takes a decade, costs billions of dollars, and the failure rate is astronomically high.
But with DRL, you digitize the lab.
Yes. The environment becomes a vast, simulated chemical space. The agent is tasked with finding or assembling a molecule that interacts optimally with a specific genomic target.
It's building drugs.
The agent searches through millions of potential compounds, predicting the 3D molecular interactions based on the genetic data. Its reward function is strictly defined by how strongly and specifically the simulated molecule binds to the target while avoiding off-target toxicity.
And it gets penalized if it fails.
Heavily penalized. It efficiently searches the chemical space to identify the absolute best potential drug candidates before a single physical chemical is ever synthesized in a real lab.
It's basically playing millions of biochemical games of chess in a simulation, moving atomic structures around instead of knights and pawns to find the exact chemical configuration that will treat a specific disease.
That's a perfect visualization.
And the text notes real-world successes here, particularly with rare genetic illnesses like Duchenne muscular dystrophy.
Yes. Duchenne muscular dystrophy is caused by a severe mutation in the dystrophin gene. Using DRL, researchers were able to simulate agents that optimize small antisense oligonucleotides,
Which are what?
tiny synthetic strands of nucleic acids. The agent's reward was tied to finding a sequence that could bind to the patient's RNA and coax the cellular machinery into essentially skipping over the mutated part of the gene, restoring partial function to the protein.
The agent literally learned the chemical rules of the environment and engineered a custom molecular patch. That is breathtaking.
It is.
The text also mentions DRL being used heavily in drug repurposing.
Drug repurposing is incredibly efficient. Instead of inventing a new molecule, DRL models analyze existing FDA-approved drugs. They cross-reference the chemical properties of those drugs with massive genomic datasets and past clinical outcomes to rediscover neglected drug disease pairs.
Finding new uses for old drugs based on an individual patient's unique genetic profile. That is a massive systemic advantage, because an existing drug has already passed phase 1 safety trials for human toxicity. If the DRL agent finds a new use for it, you bypass years of regulatory hurdles and safety testing.
It drastically accelerates the pipeline. DRL is also being deployed in variant classification, dynamically adapting the decision-making process for prioritizing which genetic variants are pathogenic as new evidence appears in real time.
Incredible.
But perhaps the most futuristic and consequential application mentioned in the text is the use of DRL in gene editing.
CRISPR, the ability to physically cut and edit the DNA sequence in a living cell.
Yes, specifically the CRISPR-Cas9 system. It is a revolutionary tool, but it has a major vulnerability. The system relies on a guide RNA, a molecular GPS that tells the Cas9 enzyme exactly where to cut the DNA strand.
Right.
Designing that guide RNA is highly complex. If it is slightly off, the enzyme might cut the genome in the wrong place, creating catastrophic off-target mutations.
So how does DRL secure the GPS?
DRL is being used to refine the guide RNA design over several simulated cycles. The agent proposes a guide RNA design, the environment simulates the cutting process and the complex cellular repair mechanisms, and the agent is rewarded for high on-target precision and heavily penalized for any off-target activity.
It learns the best route.
It uses ongoing feedback to achieve the absolute most precise genetic alterations possible.
It's acting as a highly advanced flight simulator for gene editing, practicing the molecular surgery 1,000,000 times virtually to perfect the GPS coordinates before making a single permanent cut in a real human cell.
Exactly.
But as incredible as DRL is, the text makes it clear that the future of this entire field requires us to break down the existing silos in biology. We have to look at Section 7, the future directions of transfer learning and multi-omics.
Let's start with transfer learning because it acts as a direct structural workaround to the data-hungry problem we discussed in Section 4.
Right, where you need petabytes of data.
We established that training a deep learning model from scratch requires petabytes of labeled data. If you are studying a rare disease or a non-model organism, you simply do not have that data. Your pipeline is totally stuck.
So how do you train a model without data?
You utilize transfer learning. Instead of training a blank model from scratch, you take a sophisticated deep learning model that has already been trained on a massive, well-characterized dataset, like the foundational human genome or the genome of a highly studied laboratory mouse.
Okay.
Through that initial massive training, the model has already learned the fundamental grammar and the general physical properties of genomic sequences.
It already knows how to read the language of DNA.
Exactly. Then you transfer that pre-trained model and simply fine-tune its final layers using your much smaller data-poor dataset from the newly discovered species or the rare disease variant.
That makes total sense.
It drastically reduces the required compute time and the amount of data needed, and it helps prevent the overfitting problem because the model already possesses a robust, generalized foundational understanding of biology.
That sounds like a magic bullet for researchers operating outside the massive, well-funded institutions. But the text provides a fascinating example that shows the limits of mathematical generalization. It isn't foolproof.
It is not. The text details a specific study where researchers successfully trained a model on the human genome to detect transcription start sites, or TSS.
What are those?
These are the critical locations where the reading of a gene begins. They took this human-trained model and transferred it to mouse and chicken genomes. It fine-tuned quickly and predicted the sites beautifully. The model generalized across the species.
Okay, so far so good.
But then they attempted to transfer that exact same model to a zebrafish genome.
And what happened?
It failed completely. The predictive accuracy collapsed. The generalizations simply broke down.
That feels counterintuitive. If it works for a human, a mouse, and a bird, why does it fail on a fish?
DNA is DNA, right? DNA is the same medium, but the evolutionary distance between the species was simply too great. The complex molecular mechanisms and regulatory grammar governing transcription in a zebrafish are fundamentally less conserved relative to humans than those in a mouse or a bird.
So they evolved differently.
Millions of years of divergent evolution altered the underlying biological rules.
So the AI learned the rules of human biology, and those rules applied broadly to mammals and birds. But the fish was operating on a slightly different biological operating system. The mathematical model assumed a pattern that simply did not exist in the physical reality of the fish.
Precisely. Deep learning is an incredibly powerful statistical engine, but it cannot magically override fundamental evolutionary biology.
Right.
If the underlying mechanism has diverged through evolutionary time, a mathematical model trained on one branch of the tree of life cannot blindly predict the reality of another. It is a stark reminder to researchers that we are modeling physical, evolved biological systems, not just manipulating abstract numbers in a vacuum.
Which perfectly transitions us to the final and perhaps most expansive future direction discussed in the book, multiomics. We have spent this entire deep dive mostly talking about genomics, the DNA sequence itself, the ATCGs. But as we established in the very beginning, DNA is just the static blueprint.
Yes. Genomics alone is entirely insufficient to understand complex biological functions, systemic diseases, or aging. You have to integrate genomics with the other dynamic layers of the cell.
Which are?
You have to integrate transcriptomics, the RNA transcripts, proteomics, the actual 3D proteins built from the RNA that do the physical work in the cell, metabolomics, the chemical processes and byproducts, and epigenomics, the structural modifications that dynamically turn the genome on and off.
I want to frame multiomics for you, the listener, by expanding on an analogy. Relying purely on genomics to understand a patient's health is like reading the script of a play. You know the words, you know the characters, you know the general plot.
It's static.
Right. But multiomics is actually going to opening night. It is watching the actors perform the script. It integrates the stage lighting, the swelling music, the improvised reactions of the actors, and the audience gasping in the dark.
I love that analogy.
If we apply that to a clinical setting, relying just on the DNA script might tell a doctor you have the genetic predisposition for a specific tumor. But the multiomics opening night tells the doctor whether that tumor is actively growing right now, what specific protein it is currently using to hide from your immune system, and how your metabolism is reacting to the stress.
It's the full picture.
Multiomics gives us the complete, living, breathing picture of the biological system in action.
And deep learning is currently the only analytical tool capable of integrating all of that heterogeneous data at once. Researchers are building multimodal deep learning networks. These architectures are fascinating.
How do they work?
They have distinct parallel branches. One branch processes the high-dimensional proteomic data, another processes the time series RNA expression data, and a transformer branch processes the sequential DNA data simultaneously.
All at the same time.
Then a shared central network layers and integrates all those different representations to produce a single holistic prediction.
It's mapping the entire biological ecosystem simultaneously, looking at how a change in the DNA ripples through the RNA, alters the protein, and disrupts the metabolism.
It allows us to find novel systemic disease processes that you would never see if you only looked at the DNA sequence in isolation. It is the ultimate realization of the architectures of perception.
We have covered an immense amount of ground today. We started with the paradigm shift from physical data collection to the crisis of pattern recognition. We explored how architectures like CNNs, LSTMs, and self-attention transformers act as the new tools of perception, finding biological truth in petabyte-scale noise.
Right.
We looked at how they are solving the billion-piece, error-ridden puzzle of fragment assembly and moving us past the era of computational exhaustion.
But we also rigorously examined the systemic realities and constraints of the pipeline. The fact that the ultimate promise of personalized precision medicine is entirely dependent on a structural data model institution pipeline.
It's not free.
No. These tools require vast compute power and massive labeled datasets, which concentrates predictive authority in specific institutions and introduces the mathematical risk of out-of-distribution failure for underrepresented biologies. The tools are undeniably getting sharper, but we must remain incredibly vigilant.
Absolutely.
We have to continually ask who builds the datasets, who controls the computing power, and crucially, how we navigate the opacity of the black box.
Because better mathematical prediction does not automatically equal better biological understanding. We have to utilize tools like shape and LIME to force the models to translate their logic, ensuring we understand the biological why behind the algorithm's what. And as we move into the era of deep reinforcement learning, finding novel drugs in virtual simulations, and multi-omics mapping the entire living theater of human biology, the clinical stakes have never been higher.
Deep learning is undeniably a game changer for biological systems. It is fundamentally shifting how humanity interacts with the building blocks of life, but we must always remember that it is a tool. It is a mathematical system with constraints, limitations, and structural dependencies. It is not magic.
And that brings us to the end of our deep dive today. But before we sign off, I want to leave you with a final thought to mull over, something to chew on as you process all the systems we've discussed today.
Okay.
We talked about how researchers are using deep reinforcement, learning these incredibly complex, opaque models to refine and optimize CRISPR-Cas9 gene editing. We are using algorithms whose internal mathematical logic we don't fully understand, but whose predictive accuracy we trust, to design the molecular GPS that alters our actual DNA.
It's a profound thought.
So the provocative question is this. If we are handing the design of our own permanent genetic alterations over to black box models, are we still actively guiding our own evolution? Or have we officially handed the steering wheel of human biology over to the pattern recognizer?
That's the question we all need to answer.
Thank you so much for joining us on this deep dive into the architectures of perception. This project is free and publicly accessible. If you'd like to support the time, tools, and research behind crisis in perception, you can become a supporter on Patreon.
For a shorter visual overview of this topic, see the mini explainer linked in the description.
If you enjoy this kind of systems-level analysis, please like, subscribe, and comment with books or topics you'd like us to explore next. Keep learning, keep questioning the systems around you, and we will see you next time.